otherwise known as proteinaceous infectious particles, or infectious proteins if you like to think of it. They are not only infectious, but they are known to be heritable. You can get it through certain types of genetic mutations, and they're also known to be spontaneous because what they are is basically misguided three-dimensional structures of proteins, so it can form spontaneously according to certain types of research. So what would be some examples of prion diseases? Well, prion diseases, these are just some of the things that we hypothesized. Was scrapie, which is a neurodegenerative disease of sheep. Mad cow, which if you paid attention to the news, you know that that is something that infects cows and causes literally holes in their brains. Infectious Creutzfeldt-Jakob Disease. And lastly, Kuru, and this is, we'll go into talking about Kuru much later. What they all have in common, I guess, is that they all have very similar symptoms, such as fatal insomnia, uncontrollable bouts of laughter, incontinence, coordination disorders. So I mean, it does show the signs and symptoms of something that's affecting the brain, certainly, but they are thought to be slow, neurodegenerative diseases. And this is, of course, a very broad term. All of these have different incubation periods and different times. So let's just kind of go into talking about how they work. They all work in the basis, at least from an intuitive standpoint of how this is based on the PRP protein. Now, we don't really know exactly what this does. The brain is one of those things where if you look at a textbook, you're going to see a lot of the words may being used here. So they think that it may play a role in cell-to-cell adhesion and intracellular signaling, which helps neurons communicate with each other. But they have done experiments with mice that have had without the PRP protein, and they were phenotypically fine. They were doing okay. Now that may be different for humans, but at least with mice, they have the PRP been modified at least not to have the PRP, and they've been quite functional and not really any noticeable differences. So I guess how does it work? Well, prions, I guess I'm going to go ahead and call them over here. If you know anything about protein folding, you know that they are evil versions of chaperonins. As you know, that those things that help guide a protein into its normal, appropriate three-dimensional conformation. Well, and this is all, we're talking about the PRP protein here. PRP in the brain. This is the normal conformation over here. This is something that, without really going into a whole lot of, I guess, biochemistry on protein folding, this is an alpha helix. At least an alpha helical structure as the base of it. So once these evil chaperonins, these prions come into play on this PRP molecule here, or this PRP protein, sorry, it is converted to this beta sheet. And as you know, that these are all secondary level structures of proteins. So this conversion here can actually cause not only the misfolding of other certain types of proteins, but can cause plaquing to build up. So this beta sheet can come in and it can take another alpha helix and then convert it back into a beta sheet. These are all just the PRP proteins here. Not only that, but beta sheets can link together, which forms a plaque up in the brain. Plaque inside the brain, and if you have a lot of non-functional proteins clumping together inside a neuron, then that neuron's not going to be able to do its job, and that's when you're going to start having a lot of the cognitive defects and pretty observable signs and symptoms. Okay, so I guess the real main basis behind where a lot of this comes from is by a guy named by the name of Stanley Prusiner. And I hope I'm pronouncing those names right. I'm really bad at doing that. And what made his work so, I guess, impressive was that the fact that he demonstrated that treatments designed to destroy nucleic acids did not affect any of the infectious ability of the agent. I should also clarify that he used a pore/filtration methods to rule out the risk of it being a bacterium. Okay, so we know he knew to kind of move on to whether or not it was, say, like a virus or anything that could be causing any anomalies in this case. So he demonstrated this by treating it with something that would destroy the nucleic acid of something, and that didn't really do anything in terms of stopping the infection. So what else would you move on to? He treated it with certain protein denaturing agents, one of them being phenols, lots of types of proteases, and in doing that and just attacking proteins, the infectious agent, was inactivated. And this won him the 1997 Nobel Prize, because prior to this experiment, everybody had thought that you needed a nucleic acid to be infectious. You had to have something to give yourself an especially if you're going to be inherited from one point to the next. If something, we're going to follow along on Cook's postulates here. If I'm going to take something from this guy, I'm going to go around and I'm going to, purify it and then inject it into someone else. Well, that means that it has to replicate. So I guess the problem would be, how could something replicate if it's made of only proteins? And that's going back to when we had talked about the alpha helix and the beta sheets, something that's really unheard of at the time. There was a bit of a controversy, however. Or not necessarily a controversy, I would say, so much as there was a disagreement among certain people in the scientific community. Because from a molecular basis, it makes all intuitive sense. There's I guess a gap between what you can see in a lab and experimental data and what you can see in from a molecular basis. Because remember, Cook's postulates is kind of limited into how well we can test that postulates. So one of the possible, I guess, alternative descriptions for a lot of these neurodegenerative diseases would be heavy metal poisoning. The pathological implications of PRPC, this C here just stands for the normal PRP protein, or normal in this context, it means cellular PRP, basically saying that if you oxidize any type of protein, whether by consuming, say, metal compounds, depending on, I guess really you have to look at the nature of the patients themselves, but anytime you're put under an oxidative stress, this can cause damage to your protein's three-dimensional structure, which can cause a normal, healthy protein to be converted into something that is, non-functional, that is obscure. And that's what this SC here stands for. We call that the scrapie, which is just the name that we have given to proteins that cause prion-like diseases. Okay, so I guess the thing that it throws me off a lot of it at times is that prions aren't really well defined, and I wish they would make a difference between whether or not something is infectious or if it's caustic to oneself. Because, for example, yeast, have misfolded, seemingly non-functional at the time forms of it. But the thing is that it actually helps them. They grants them a resistance to antifungal agents, and so they'll actually increase this conversion into this three-dimensional structure upon exposure to certain antifungal agents. So say that I'm a yeast here, and I'm going to get exposed to some type of a fungicidal agent, and I can make basically a secondary wall structure of protein based off of prions that'll protect me from that. So whether or not they're actually just misfolded proteins or just proteins where it's I guess changing from one to the next rather than breaking it down and making a new one, who knows? It certainly would be much more metabolically efficient to do that, but that's what is the case in yeast. There's also a Nobel laureate by the name of Eric Kendall, and I hope I'm pronouncing that right as well, who did some really interesting studies on prions and memory formation. Now, again, I'm not talking about PRP here. I'm talking about a three-dimensional structure protein that had its three-dimensional structure changed by some unknown pathway that we're not quite sure about. But anyways, what he showed is that animals make and use prions in their nervous system, as part of their nervous system for an essential function of stabilizing synapses that constitute long-term memories. It's actually kind of difficult when you think about how neurons can stay, so well connected for so long. I still remember how to ride a bike, yet I haven't done it in a very, very long time. I still remember all the combos to Lord of the Rings, the video game, and the PS4 just came out. So this is something that I thought was really, really helpful to show that they're not all bad. So once the prion's chain reaction, I guess for memory function, gets started, it's self-perpetuating, and thus the synapse can be maintained after time and the initial trigger is gone, perhaps for even a lifetime. But what we don't really know is how this thing right here, how first the prion is triggered or why it only happens in certain synapses and not others. But the thing that I really want to explain is how would something like that happen if it's just a protein that gets misfolded? I mean, if it's observable in multiple, animals, multiple types, both mammals and I think Drosophila is what he exposed it on, then it's clearly an advantageous mechanism. So anyways, moving on. Keep in mind, yes, which we talked about later, that memory prions are not the PRP protein and however do not exhibit any type of an infectious nature, which again, I think we need to get a little bit clearer definitions on what exactly a prion is and what exactly a prion isn't, what the difference between a prion is and a misfolded protein, which we haven't quite, I guess, distinguished those two. All right, so now I really do want to talk about something that is, I consider extremely important, and this is the story of Kuru. So in the late 1950s, early 1960s, there's a Papua New Guinea tribe of people that are coming down with this medical illness, and the locals had called it Kuru. And what it was basically very similar to what we had seen with mad cow disease and what we have seen with scrapie is, I'm just going to turn it here, neurodegenerative disorders. And the NIH had sent a man by the name of Dr., and I hope I'm pronouncing this right, Gadusik, I don't know how you pronounce his name. I'm just going to call him Dr. Gaj for now, who later ended up being convicted of child molestations, but this is just a good story that we should all keep in mind, and something that's not really talked about a lot in textbooks for some reason. So anyways, he had hypothesized upon observing that the disease is being spread by means of something infectious. He thought at the time it would be something viral that was spread by cannibalism rituals involving the consumption of nervous tissue, eating brains. Which again, kind of makes sense because remember where PRP comes from? Well, it comes from brain tissues, neurons. But the thing that was really interesting was that the patients that were infected, they didn't show any signs of inflammation or fever or any type of immunity to anything. That if it were a prion, they'd be helpless against it. So kind of confusing, and he didn't really do a whole lot to rule out anything else, which is what we're talking about here, without taking any precautions to rule out, say, toxic, genetic aspects, nutritional deficiencies. Keep in mind that when you're talking about the third world, these things can crop up. He reported to the NIH and formulated that a slow virus, which was, keep in mind this was the 70s, as the causative agent of his disease. So having failed to, try and go following through with Cook's postulates and isolate/purify the virus, he went straight to animal experiments to try to see if he could prove that it was infectious. And yes, I am giving a hindsight is always 20/20 look at this, but it's important that we really analyze this incidents from happening because things were blamed for it that weren't causative agents. So animals that had been injected with blood, urine, and even the cerebrospinal fluid, nor monkeys who had been fed brain samples of Kuru patients failed to show any similar signs or symptoms, which is kind of odd because it's not like a virus where we're talking about a host range here. It's just a misguided protein. It's going to cause disease in a sheep, it's going to cause disease in a cow, and it's going to cause a disease in you. So only after, this is kind of disturbing the, I guess, extent that he had to go through, surgically injecting homogenized nervous tissue into the chimpanzee skulls did Gajus see any observable change in terms of neurological abilities. But the thing is, cases varied from, coordination problems to full-blown encephalopathy. So again, kind of, that's always going to be harmful regardless of whether there's infectious agents or not. So after physical and then chemically treating, trying to remove any of the infectious agents, and no signs of foreign nucleic acids, which again does kind of point to the idea of it being a prion disease, he announced that he was dealing with an unconventional virus, that this was something, that the CDC had found something new that they needed to stop immediately. And so in 1977, he won a Nobel Prize for a virus that he had never found or even followed through with Cook's postulates. But because I think that just the severity of the cases and the way, how out there they really were, he won a Nobel Prize for that, which is kind of weird. And he was actually questioned afterwards to show any evidence that he had seen that they were performing lots of cannibalism type rituals, and he never released them, stating that they were too offensive. Now, according to certain books that I've read, he actually did release them, but would later admit that those weren't actual cannibalism photos, those were just villagers eating things, which I felt was kind of obscure. And so here's where one of the few times, I guess, in my career where I've actually thought that a biologist was full of crap and completely sided with an anthropologist by the name of Lyle Steadman, challenged him, saying that when he had gone there a couple of years prior, he hadn't observed any type of cannibalism rituals, especially for that of nervous tissue during his 2 years spent in Papua New Guinea. Nor did he see any evidence of it during his time, which was, again, prior to Gajewski's arrival. So I guess if we were to kind of look at this from a, So I guess if we were to kind of try to look at this from a standpoint of just things that doesn't quite make sense to me, cannibalism is a part of our history, a part of the history of man. Whether you're go to like the Aztecs in Aztecs in parts of Mexico and South America, certain parts of Africa, and even up in Neanderthals, cannibalism is a part of our history. We've done that for a long time. So all of a sudden, a very old, very well-established ritual would all of a sudden cause infectious agents to be spread about from one to the next, especially whenever an anthropologist is saying that it hasn't been there. He never observed any type of cannibalism. And even Dr. Gages himself never admitted that I saw cannibalism. So I think it's kind of disturbing to me that the scientific community has often gone, and there's lots of people talking about it, hankering, about how the cannibalism tribes have been responsible for this. I don't think that they are. My personal opinion, I don't think that this was a prion disease, and I don't think that they were eating human nervous tissue to begin with. And even if they were, that's something that's been a part of human evolution for a very long time. So just a lot of iffy there. If you ask an anthropologist about this, they're probably going to give you a different opinion about it. Anyways, so after that happened, after he won his Nobel Prize for essentially a virus that he had never found and what would later be blamed for prions because he couldn't find any type of a virus for it, he was charged with child molestation in April 1966, pleaded guilty, and then later on died 10 years later. So if anything says that he has integrity, it's the fact that he was willing to rape children. Good on you, man. Okay, so just kind of some last second thoughts about prions and things that I don't quite understand. So being a misfolded protein, like a prion, it would originate from humans and cows, such as in mad cow disease or Creutzfeldt-Jakob. That three-dimensional structure alone should be within the cell, themselves. Therefore, why is it resistant to extreme heat? A lot of people say that prions are virtually indestructible. And I'm going to write that. Indestructible. Because they had exposed them to a lot of pretty hardcore agents, really cooking them to try to get rid of any of those agents, and they said that it wasn't working, that they were still getting sick from this. I understand that cysteine does form a very strong disulfide bridge, but let's just talk about all the things that come into play for protein folding, okay? So you got to have the right pH, you got to have the right osmolality. You got to have the right temperature. And on top of that, you got to have, then you have to have chaperonins and things like that. And if all of these things come into play with protein folding, and they're designed, those proteins are optimal inside of me or inside of a cow, then why would heating it to, say, something that would cook any other normal protein and denature it, why are they resistant to that? Especially if we're talking about PRP, scrapie form, which we said earlier was a beta sheet, and the primary, what's holding that beta sheet together is hydrogen bonds. Now, I don't know how much chemistry you've taken, but a hydrogen bond is a weak, it's not even a bond, it's an intermolecular force. So this is something that we could easily disrupt. I can disrupt hydrogen bonds by taking this bottle of water and putting it under some, a stove, or put it in the microwave for a couple of minutes, and it'll be converted into steam. So not really making a whole lot of sense to me there. It's just some of the questions that I've never been able to find answers to. Another thing that I never was able to find any sort of answer to is how do prions cross the blood-brain barrier? So you got to think about this. If I'm eating a nervous tissue consumption, that I can understand through like improper sterile, like surgical procedures that were done, yeah, that's going to definitely integrate something into the body. But if it's something that's transmittable by food, by consuming even nervous tissue, then it has to get from my mouth, through my stomach, through that hydrochloric acid environment, through all the carboxyl peptidases and enzymes that I have to digest proteins themselves, stay in that three-dimensional structure, and then get into my bloodstream through my liver, and then all the way up to my blood-brain barrier, and even then it has to cross that blood-brain barrier to get into my neurons and to be infectious. So Again, I haven't been able to find a whole lot of information about that. I don't know if there is studies done or if not. And then something else that my book doesn't say, Wily Microbiology, I hate you, is the difference between, say, a misfolded PRP protein and the infectious agent of prions. Because I think that those are different, maybe, or are they the same? Because again, beta sheet is held together by hydrogen bonds, which makes it very, very weak. It would be denatured upon anything, you know, even the acidic environment of my stomach. But they don't really dissociate between the two. Some people say that this is the same thing as prions, and I've just heard different things, and I would really like if I could know the difference between the two, but I can't. So anyways, that's that.